Let's take a look now at some histology of these receptors. These images are images I just pulled off of Google, so you can do the same when you're studying. You can find labeled or unlabeled images of all the histology that we are talking about today, or our modules also have some good examples. So any of these are good candidates for PowerPoint questions. What we have here is a slice of skin. So to orient ourselves, let's go ahead and find that basement membrane. So up top here, we've got tons of cellular epithelium, or epidermis in this case. So if you look closely here, we've got layers upon layers of that flattened, simple squamous epithelium. That's how we know that's our top layer of the skin, stratum corneum. If we go deeper, we're still dealing with cells upon cells here, so we're still within that epidermis. And then we see that texture change. So we've got this wavy pattern here, separating the cellular part from the underlying connective tissue. That's how we know this is going to be our basement membrane. And then everything deeper is that dermis layer. Now that we have located that basement membrane, we want to look within these dermal papillae, these finger-like projections. And what we're looking for is kind of almost a thumb-shaped structure, which is a receptor, in this case the tactile receptor or tactile corpuscle, for that light touch. And so we don't see anything here. This is just connective tissue, but we do have this round structure here. It's almost like a cocoon, if you want to think of it that way. And then we have another one on the right, another cocoon shape. So these are going to be our tactile corpuscles. We know that because they're sitting superficially. They're just underneath that basement membrane, underneath that epidermis, and they're tucked within these dermal papillae. Let's contrast that now with our deeper corpuscles, our lamellar or our Pacinian corpuscles. So let's reorient. This is a much more macroscopic scale. We're looking at maybe 4x magnification of this slide. So up top, I'm just going to put an arrow towards it. This is going to be our epidermis. We still have our basement membrane, so don't get it twisted here. This is not our basement membrane. This underlying wavy pattern is going to be our basement membrane. And then everything else we see here is either dermis, put a D for dermis, or we have hypodermis. You can see this layer can be pretty thick depending on how much subcutaneous fat we have. So now that we've located our layers, we want to look within the hypodermis and the dermis for those Pacinian or lamellar corpuscles. And right away, we have a really nice example here. You can see it has these concentric layers or lamellae. It does look like a fingerprint. So that gives us a good clue that this is going to be that Pacinian corpuscle. We have another one here. And they won't always be perfectly round, but we have some really beautiful examples of it on this slide. So pay attention to these things. Make sure you go through that process of orienting yourself before you try to identify anything. A lot of times that's where students get tripped up. Next up, we have our gustatory receptors, our taste receptors. So these are things that we will find in the region of the tongue. And they have a pretty simple name. There's no fancy term. We call these taste buds, where sensory information from the food we eat, liquids we consume, is going to be sent to our brain for processing. It's that simple. Now, taste buds can be a little tricky because they sit on top of this structure called a papillae. And we just saw this term, dermal papilla. In this case, it's just called the papilla when we talk about the tongue. So let's go on to our histology slide because we don't have a model for the tongue, but we can look at lots of microscope questions. What we're looking at is probably a 40x magnification of the tongue. So you're not going to be able to tell this is tongue tissue unless you know what the pattern of papillae looks like. So basically, the tongue is made of these adjacent papillae. This is one papilla. It's a little hard to see. And next to it, neighboring it, we have a different papillae. On the other side is the start of another papillae. So altogether, we can see 1, 2, 3 papillae. This is what you should expect to see on your slide. It's not going to look like a tongue, but if you know what to look for, if you know what one of these papillae looks like, you'll be able to identify stuff no problem. So the papillae are going to be those raised structures. We have a couple different types. So we've got circumvallate, fungiform, and foliate papillae. They have different shapes, and some of them have taste buds, others do not. You don't really need to know that for the lab exams. We're only going to focus on the papillae that do have taste buds. So once you have located your papillae, you want to look a little closer. So we're going to be looking kind of on the edges of the papillae here, where they're touching each other. And lining the sides, we're going to see these round, almost onion-shaped structures. So we're looking in this region here. And we're going to have lots and lots of these little circular units. To me, they look onion-shaped. They might look different to you. We have them on the neighboring papilla as well. So here's a taste bud. So each of these circled units is a taste bud. And they're responsible for our sensation of sweet, sour, bitter, salty, and umami or savory. So altogether, make sure you can identify the papilla. In this case, there's no other descriptive term. We just call it a papilla. And be able to identify the taste buds a little deeper, kind of on those adjacent sides of the papilla. We have another example. So to study this, I just recommend pulling up many different examples of taste buds so you can get used to them. The reason you want to practice with different slides is because we do have different shaped papillae. Depending on the type, it's going to look more mushroom shaped. It might look more block-like this one. So make sure you're comfortable with the different types of papillae we might see with taste buds. Now this slide has been pulled directly from the modules. First, we want to identify our papilla. So we've got one papilla here. This whole thing is one papilla, and then we have a neighbor papilla. Once you do that, you want to look a little closer. And one thing to point out here is we're also dealing with a basement membrane. So if you're trying to find your taste buds and you're struggling, go ahead and look for this basement membrane. It still has a wavy pattern, just like we saw in the skin. And then our taste buds are usually tucked just deeper to that basement membrane. So you've got one here, this is another one, this is another one. So we're looking for that round or onion-shaped pattern to identify these guys. This one is fair game for microscope or a PowerPoint question. However, we do not have any models for the tongue.